Hi everyone, my name is Ruben and I'm one of the software developers at DArT. Thank you all for joining us for the last information session of the conference. We're very excited to show you a new development here at DArT, which is the OneDART Engagement Hub and its associated applications. At DArT, our mission is to change the world through sustainable use of natural resources. For years, we've been doing this by providing our customers with high-quality genomic data from processing in the lab to analysis and support of scientists and industry. And probably a lot of you are our customers from this era. The tools we've developed to do this are powerful. What we want to do is put these tools into the hands of our customers so they can accurately track their samples, the sample collection, and perform comprehensive data discovery and analysis themselves. And to do this, we're developing the OneDART platform. In this presentation, we're going to walk through the Engagement Hub, EcoLog, and some of the analytics tools in the context of real-life challenges and needs of ecologists in the field. And we're going to follow along with the needs of ecologist Jason Dobry, who is chasing dragons in the form of Veronica Rosenberg, or Rosenberg's monitor lizard. The OneDART Engagement Hub serves as the entry point to the OneDART suite of applications. When you first visit the hub, you'll see a login page. Everything is done securely through KeyCloak here. If you already have a DART login, perhaps you're already one of our online ordering customers because you've submitted samples to DART, you can simply enter those credentials in and you'll be ready to go. This will automatically link all of your current orders and your historical data. If you don't have a login, don't worry. You can just click register at the bottom of this, fill in a few forms, and then you'll get a registration email. And once you've finished registering that, you'll end up in the OneDART Engagement Hub landing page. This page here is called the Cloud Management page. Many of our OneDART applications require a whole lot of computing resources, and those all run in the cloud. For that reason, we created dedicated compute nodes that act as your individual machines within AWS. This isolates your data for security reasons and allows you to apply your budget only to your own analysis requirements. These nodes can all be managed from this page. You can start them and stop them and budget their usage as needed. At the moment, I don't have anything running, but I'll change that. You'll also see up the top here, I have one compute credit available. This allows new users to explore the system and experiment a little bit without actually having to invest any money. So we'll go ahead and provision by the instances here. And when we provision an instance, we have to select first which service. Currently, we have EcoLog, dartR, and dartSoft, and we'll talk more about those each in a little bit. Then after that, we'll select the compute size, which is set to medium here. Of course, larger compute nodes are more powerful and will run analyses faster, but they also cost more to run. In practice, the cost is actually pretty small. We can see down the bottom here. It's typically less than 10 cents per hour per node. Even so, we do encourage users to keep their usage inefficient, to minimize costs, and also to reduce the environmental impact. Part of the reason I'm demonstrating this via PowerPoint slides rather than live is that this provisioning process can take a long time for a new node, usually between about 5 and 10 minutes. So here I've already spun up 3 nodes. We have dartSoft 14, we have dartR, and we have EcoLog all ready to go. And these are on, and I can stop them and I can schedule. We'll get to that in a little bit. This is the cloud management page. Next, I'll look at the launch pad here. And this is where we can access all of the applications that are within the OneDART ecosystem. The first launcher, Analytics Plus up the top here, is itself a suite of additional tools. So that's where we'll find things like dartSoft and dartR and some other things. We also have EcoLog, which we're going to talk about next. We have things like a barcode generator for creating unique barcodes. Order services here is our online ordering service. So this links through to all of our DART orders and DART sequences. The storage is where you'll find all of your data, all of your historical orders, and the results of the analyses that you run here. Also on the right here, you'll see a bunch of user guides. So we've got user information ready on that same web page. Here's a few screenshots of some of the applications. Up in the top left, we have EcoLog. In the center here, we have dartR, which hopefully you had a little bit of a look at already. And in the top right here...
You are muted.
I am muted? No. In the top right, we have the online ordering portal, and I'll go to that just to show the compute credit. That's where we'd go to request more compute credits. So our $1 of compute credit would give most of a day of experimentation. But if you're doing some serious analysis and are going to be leaving things running for a while, you'll need a little more than that. Getting compute credits is super simple. We just put in the amount that we want and hit checkout here, and that will raise an invoice. And you would pay that exactly the same way that you'd pay for a DART-seq analysis or something like that. But by allowing people to control their own compute credits, we're trying to keep everything as transparent and user controllable as possible, so that people will only be paying for the computing resources that they actually need. The idea is that when you start the computing nodes, you use them and then you turn them off when you finish, so they're not consuming resources when you're idle. It is very easy to leave at the end of the day and forget to turn this off, or even leave it over the weekend and get a bit of a cost overrun. Ask me how I know. So we have a scheduler in here as well. So you can actually have these nodes automatically turn on at a certain time or on a certain day. The way I tend to use this is I just have a blanket turn off at just after 7 p.m. So if I've forgotten to turn it off at the end of the day, it'll still turn itself off. At this point, we want to dive a little bit deeper into some of the actual applications, and we'll check in with Jason and talk about how we apply this to EcoLog.
Hello, everyone. My name is Jason Dobry, and I have been an ecologist for over 20 years. My first field experiment was back in 2003 with rattlesnakes in Washington State. I was an undergraduate and got a grant to do the work, but I didn't have a lot of guidance, so I had to figure out on my own how to do everything. We didn't have all the great apps like today, so everything was paper-based, and lots can go wrong with paper records. You get illegible notes from collaborators and volunteers, things get lost, things can get ruined by driving your Land Cruiser into a river. Bad weather can blow things away or ruin your notes. And when you're looking for rare or secretive creatures that may often even be dangerous themselves, there are situations that often occur more often than perfect collections. And you know, many failures, you find nothing. Time and funds are always limited. Environmental conditions impact collection efforts, so it might be a perfect day, you have to go right then. You're often unprepared for those events. When you finally have some level of success, adrenaline is in action. You might be dealing with dangerous animals, venomous reptiles, so many unexpected events. So fast forward, that was a pains that I went through in my first experiment. And then fast forward to my PhD in 2018, where I knew I wanted to be as digital as possible. but there was still no clear solution. So I began using a series of apps that I used to collect the data, but these were spread out and led to other series of problems, such as maybe missing metadata or lack of unique sample IDs, inconsistent data entry, if different gaps in the spellings of things and that kind of thing. And then heterogeneous data types where you have GPS coordinates and sensor data or photographs and specimen measurements all in different places. And so data fragmentation becomes a big issue. And then you also have version control problems. And so I often dreamed about a centralized application that would take away all these pains of data collection. Handwritten notes are a thing of the past, and so here we can, in the OneDArT platform, we've came up with a solution that integrates all our data types. We integrate our GPS coordinates, and all our everything is barcoded, and all the samples are tracked easily. There's no issues with different spellings because we have drop-down menus. The data types are all centralized, and so you don't have the fragmented data storage, you don't have version control problems, and this is the solution that we have come up with and that we're introducing you to. And so Taylor will be able to take you on a journey through the web application, and then I'll walk you through an example with the mobile app.
Hi everybody, I'm Taylor. I'll just share my screen quickly. It's my screen. There we go. Hopefully you can see that. Yeah, so I'm Taylor and I'm part of the OneDART team here at DART. Just to follow on from Jason's explanation of the work he was doing, I'm going to show you how we designed EcoLog as a consolidated platform to collect data for a study like the Heath monitor study. So when we started, we got together with ecologists such as Jason and others too from a diverse set of backgrounds. And we designed EcoLog as this data collection solution that's integrated into the OneDART platform. So we're bringing together the functionality of multiple applications or even of pen and paper data collection all into one platform. So what is EcoLog? It's essentially two applications that are integrated. We've got the mobile app for data collection in the field, and we've also got the web app. The web app's for configuring projects, customizing data collection forms, organizing samples to send them to DART. And we can also create dartR metadata files with them. So the web app is where I'll start the demo. So I can access EcoLog from the OneDART customer portal, the launchpad that I've got here. So it's just one of these apps. And here we've got the dashboard. So the dashboard, we can see a map preview of some of the data that we've collected. We've got a survey here and various data collection points. And then we've also got our various projects that we've been working on the left here. And this one at the top, the Heath monitor study, that's got some of the data that Jason had used for his work on Heath monitors. So in Jason's example, the study started with a survey. It's basically find the location of monitors. And in EcoLog, you can do this with a survey. So we've got this walk path here. As you can see, you've got the walk path, then some observation points along the way. Once he determined that this was a good area to focus his study efforts on, just Namadgi National Park, just south of Canberra. Once he decided this was the area that he wanted to do the study, we can use EcoLog to set that all up. So next I'm going to show you the kind of next phase of the study, which is setting up the track collection and doing all the admin before we get into the field. And then we can start by looking at the records we collect. So EcoLog, it has a large variety of records that can be collected. And we group them into two categories. So we've got the administration records, and these things would be like the species. So for example, the species in this project just added a few different monitors, but in the middle, we've got the Heath monitor, species name, that sort of thing. And you can add a new one in this field here. Another example of an admin record would be a site. So I've added the Namadgi National Park. But if you want to add more, you can draw them on the map, essentially. Another good example would be trap types. So this isn't the actual trap themselves, but to categorize traps. So one of the main ones that Jason used was the Elliott trap, which is a kind of metal box for catching vertebrates. And so we could add a new one there if we like as well. So these admin records can be found on the menu on the left over here, and as you can see, there's a lot of options. And then the second type of record is the data collection records. So this is what we're collecting when we're out in the field. That could include things like samples, for example. We have a bunch of different types of these data collection records, and they are stored in a hierarchical record structure. So, for example, we have a trap in a trap kind of methodology. Start with a trap, and that's put out at a location. And each time that trap is checked, we've got an event is recorded. So that's the collector, they record the start and end time, any comments. If there's an animal or a specimen that they find in that event, they can add a field record, which in this case is a form that we've added with all the measurements for the monitor. And of course, if you want to take a sample, like a blood sample, for example, you can add one to that here. So choose the species, got the barcode for it. This is mostly done in the field, but we can also do it on the web here too. Let's go back to the traps. So if I go to add a trap, you can see the type of fields that we're collecting, and these will link to the admin records too. So I've got a couple of sites here, like Ginninderra, Namadgi National Park, and the trap types. So it just shows how those records can be linked together. So then we get into what fields and what data are we going to collect for our study. And what you collect with these records is entirely up to you. We do have these kind of base forms, but most people will want to customize that some way. And like Jason mentioned, one of the difficulties for him was when collecting data was the mix of data types. So this aims to have it all integrated. You can customize forms to collect images, audio, GPS, and all sorts of data types for these custom forms. And in my opinion, these forms are probably one of the most powerful features for EcoLog, because firstly, we're replacing them with paper-based recording, meaning that we can reduce data entry error, increase like storage security, that kind of thing. And secondly, we can actually ensure that the data entry is consistent. And I'll show that in a little bit too. But one thing to note is that the forms are created by record type. So this trap record type, you can see the different forms that we've got for it there. So I can look at the Heath monitor trap. So got a dartR.base trap. But I can see all of these types from the management page of a project. So here we've got, we don't have to use all of these. It's entirely up to you. In this particular project, we're using most of them. So I can go to an existing form that we've created for this study, which is the field record form. And that's what we're taking the measurements of the specimens with. So this one I can edit just to show you what the form customization looks like. So and just to note, it's very flexible with how you collect the records, but this is just how we're doing it with this particular study. And then any field records that are created, we can add samples to those. So this form shows the fields that we've created for data collection, lots of measurements and things like that. We've got the control panel on the left and the preview on the right. So this is what the preview, it shows what it looks like when you're in the field. The forms are based on JSON forms, not JSON, but like JSON format. And if we want, we can add new fields by clicking and dragging these elements. So you got that control one there. Won't save that, but I'll just give you a look at how you can kind of customize it. So there's different data types. And I'll talk about each of those in a sec. Can label, description, can make it a required field. That can be really important as well. We've got some data validation and rules and that kind of thing as well. So to give you an idea of what these data types mean, so we've got 4 of them that you saw. The first is a string, which is essentially just any alphanumeric text. In this name field, I can type something like test123, random characters. That's all good. The number is a decimal point. So that's useful for things like body mass. So if we have a monitor that we find and it's 0.98 kilos, you can enter that there. You could also just change that to grams and have it as an integer as well. But so the next one is integer, which is a whole number. I've added that for all these ones which are measured in millimeters. So the tail length of a particular monitor might be 450 millimeters long. Can add that, but it won't let us create well, can't say anything, but I'm trying to type a decimal point right now and it won't let me. Then the last one we've got is a Boolean, which is essentially just a yes or no. For Jason's study, he wanted a checkbox to show whether a specimen's tail was complete. Sometimes their tails aren't complete. So we've got this checkbox here, which we could just say whether it was complete or not. There are other more advanced ones. Those are the kind of basic 4. So we can also link to admin records, like I showed with the trap type. That's an example of that. You can do the required fields that I mentioned. We could also do lists. So this gender list, we've got male, female, or juvenile/unknown. With young monitors, it's hard to tell. But what this does is that if this was a kind of free text string, anyone that's doing it could enter like the full word, like male, or they could just type in M. Could be like capitalized differently, that kind of thing. So lists are good for just getting like really consistent data entered. And another thing to mention is that we've got data validation, which is really powerful too. So a good example on this particular form is the condition rating. So the condition rating is just rating of 1 to 5 on the health of the monitor. So 1 being low, 5 low health, 5 being it's a good condition. Now, because it's a condition rating of 1 to 5, we don't want someone to enter something like 6, whether on accident or purpose. But we can add these data validation rules to make sure that the data that is entered is consistent. So the way I can do that is just go to the settings on that particular condition rating control. We've got validation here. I can set the minimum to be 1 and a maximum of 5. So that means if I type in 6, I'm going to get an error message that lets me know I can't do that. And this is really useful for where we've got like multiple people collecting data for the same study. All right, so once this is done, you can save the form, just give it a name, a version name. I won't save that particular one, but once it's saved, you can select from the list. And of course, that goes for each record type. So we're almost ready to go out to the field and start collecting data. But one last thing that I want to show you is the barcode generator application. So you can access it from the OneDART Launchpad. And then here you can generate unique barcodes. And like in Jason's case, he would use them to put on the blood sample vials. He's collecting blood samples from the specimens. Each sample receives a unique barcode, ensuring it remains linked to the correct observation throughout the workflow. So there's a few different options there. I won't go into it too much, but now we're getting to the EcoLog mobile part. So I'll pass over to Jason, and he can show you how he'll collect data in the field.
All right. So we've been driving for about an hour out into Namadgi National Park, and we've got a trailer full of Elliott traps. Each trap has its own barcode ID, and we've got a bunch of collection tubes. They've all got barcoded information on them. And we pulled into our first spot. We are down here in this valley here along the fire trail. And we want to set a trap at a spot that we had a camera trap earlier that we had set a rotten chicken bait, and the lizard was munching on it. So we know that they're in this area, so we went ahead and set another trap here. And so the first thing that we would do is we would scan our barcode. We'd enter our trap ID. We'd select our site. And then we would move on and do that with a series of traps. So after we've got our trap set, like on this one here, we can click on it. Then we've gone through, we've got volunteers, and we've gone and we've found a goanna in this trap. So we're going to go ahead and we're going to add a record. And we're going to collect the animal. We're going to pit tag it. So then we're going to scan the barcode of the pit tag. We're going to name this animal. We're going to paint some code on its back, maybe a 4 or a D or an R or a square or a triangle or something. And then we're going to collect all this data from the custom form that we had designed in our study that Taylor had just showed you. So it's just simple. You add in a value, 567 millimeters. Tail length is 200. The full length would be 767. Body mass is 2.5 kilos. And so on and so forth. Then we're going to submit that. Then we're going to go and we're going to add our sample. So we're going to scan our barcode on our collection tube. it's going to read it here, and then we're going to enter in the species. It's Veronica Rosenberg, and then any interesting comments that we want to make, then we're going to submit it. And then we're going to submit that record. And now we have collected a sample, and then we will continue to do that. We could also have incidental samples that would go in here. Same process, except for there's no trap associated with this. It's just a barcode for collecting the sample, and then we would submit that. If we want to go in and look at our settings, we can manage all of our record levels here. So for incidentals, we can decide whether we want to have all the hierarchical fields for the field record with the incidental, or if we want to have just the field record or just the sample. So the whole thing is customizable to whatever it is that you want. And then once we're done, we would upload the data, and it goes back into the web application. And then from there, we would, Taylor will show you how you can consolidate that information, submit it to a DART plate, all in one place. And then from there, when you get the data back from DART, it will be uploaded into that, into the OneDART platform. And then you can do all the analysis from there using all the other tools that we have, DS14 and dartR. So I'm going to pass that back to Taylor so he can show you how it works with the web app for the once you've uploaded the data.
Thanks, Jason. Just share my screen again. Get a bit quicker at that. Okay, so now we've got Jason's data that's been uploaded, and we can move back to the web to view and curate it. So the data is structured hierarchically, like I said before. You can see here that Jason's got these traps for the field monitor study. And so then there's like events below that, and field records and samples, so on. So for our uploaded data, we've got this version control that's handled by the form versions. So for example, for the traps that are associated that we've got with this project, we've got 5 traps uploaded right now, but they were created under 2 different form versions. So these 3 were created with the dartR.base form, and we've got the Heath monitor trap, which we've got 2 of. So if you create another one and then go and collect data with that, there's no getting mixed up with a previous data set. As the data sets and projects grow, it can get a little bit unwieldy like that. And so what's great about this is that any user on the instance records data, and it's stored in the same place and structured in the same way, which solves Jason's problem of the fragmented data storage. We can also export these just by record type, or we can also go to the management page for a project and download the whole project. It just pumps out all in one go, but it's good for having backup solutions too. So next I'll just show you one of the samples that we've uploaded. So let's go to projects, traps, events, field records. We've got this one field record here, and it's got 2 samples attached to it. So as you can see here, every single sample, it generates a sample ID. So this one at the top is the sample reference, and these are always unique. And then this one has a barcode attached to it, so that the barcodes are added on automatically as well. This means that there's no duplicate or unclear IDs to mix up tracking when we're sending it to DART later. So these carry through if you're creating subsamples too. And like Jason was saying before, sometimes when you're in the field, there's dangerous animals, like a bit of adrenaline, just out in the bush. Maybe you forget to actually record a sample. So you can go and add it here later on as well. So in that field record for that one particular specimen, you can add a field record, just one of these barcodes, choose the species of 2 fields. So I can just create that there, and I've got another sample. But then we get to what we do with the samples. So we've got our data collected, and it's ready to plate it up and send it to the DART laboratories for sequencing. So to do that, we just go to the plates page. I've pre-prepared a plate here, the monitor plate. And if I go into the view wells bit, you can see that there's a couple samples attached to it. So when I click one of these, I can just add a previous sample, and I can't remember the reference of the one I added, but I could just add another sample there. And there, so for all your samples, you can plate them up. And anyone that's done this before will know that that once you create an online order in DART's online ordering system, you'll need to add a sample tracking file. So previously, we provided a CSV template where users could enter data. But now what we have is you can actually generate one here in EcoLog, and it's got all the data already pre-filled in there. So you can download that. Just a note as well, you can save it locally, or you could save it to the cloud. So just for an example, I'll show you saving to the cloud. So this is the integrated OneDART data cloud where you can access all your data. It's all there safe. So go to my workspace. I created an EcoLog one there, and I can just save that file. So next, I will go to the online ordering app. So to do that, I've got it there, but you can access it from here, the order services. I'll go new order. And if I want to create a new order, fish, organism, Baroness, that's the monitors. Choose the DART product and the product variant. So I go for the medium density sequencing. And here I can upload that sample file. So I've got the sample tracking file there for the plate that I created. And it's done. Then I can go next and just keep going with that. I'll just well, I won't create that. I'll just get out of there and just go back. So one other thing to mention is that once we get the data file back from DART, I can run analyses on it. And Ruben will show you that in a minute. But one last thing I want to show you is that how we can create these metadata files. So we can go to the submissions page. We've got a monitor submission that I created. Submissions are basically groups of plates. So in this one, I've only got the one plate that I already sent for analysis. But here I can generate a metadata file that you can use in dartR when you're doing your analyses. So here, if I just, hit across. So here I've got my plate IDs, rows, columns, species, lat longs, that kind of thing. So, so obviously this is used in dartR in conjunction with the analyzed files. So in conclusion, that's the end of our section on EcoLog. Just to recap, we got the EcoLog web to manage our projects and customize our data collection forms. The mobile is the digitized data collection app. And then we can use that data to create sample tracking files for DART's online ordering system, and also link those samples to analyses with the metadata files. So now I'm going to pass on to Ruben to discuss DS14.
Thank you, Taylor. Do a quick screen share here. Okay. Thank you. This is Ruben again. I'm going to rejoin the process, imagining that we have done our submission to DART for DARTSec or a similar process, and we've gotten our order back. That is in our storage. Let's go to the, well, actually, what I'm going to show you now is the DARTsoft 14 application within OneDART. This is part of DART's secondary pipeline. Once samples have been sent to DART and sequenced, we previously ran this DS14 computation internally to provide the reports and the data that we supply to our customers. Since this is very dependent on the actual samples that are within that selection, if somebody gets some more samples in, they would need to request a co-analysis. But now, with putting this into the OneDART platform, we want to put this into the end user's hands, so researchers can reanalyze their data with different parameters, perform co-analysis with subsequent orders that come in, or data from new collaborators, or particularly as an iterative process, depending on the results you get from dartR. So you can get your dartR results and then go back and do different kinds of analysis on your core data. So if I go into my Analytics Plus Launchpad, I can see the applications I have there. And we'll go to DARTsoft 14. And we'll quickly launch an analysis. If I hit Browse Orders down here, that will bring us to our cloud storage. Let's have a look into my data. And here is all my orders. And right down the bottom of here, I have a couple of Varanus orders. So let me just select two of these because I want to do a co-analysis. And add these to my analysis. I get plenty of steps to add more orders or remove things, but I like what I've done there. This will pull in all the extracts that are within those orders. And I'll choose my extracts. There's plenty of ways to display this and filter here, but I chose these ones because there's not a huge amount of extracts within these. So I do want to make sure that I'm choosing them from across two different orders. So this is 3987. I'll choose 3 from that, and 3 from 4360. And 6 will do for my example here. Again, a chance to check that I've got everything that I want. I have to do an extract name, extract list name here, because I want to keep a good record of exactly what I put into this analysis. Okay, now I can just run this straight away, but there is also an extended parameter list. And this is something like an expert mode. But I am not myself a geneticist, so I will stick with the default values for all of that. And I'll name this experimental analysis. Double check everything, and I'll submit that. Now, even with only 6 samples in there, this will probably take 5 to 10 minutes, probably on the lower side of that. But we can see this running here. I can look at the logs and actually see the processes that are happening in the background. But instead, I'll just go to completed jobs here. And here's a few that I've done previously. I'll just view my outputs. And again, this is having a quick peek into the cloud storage. It takes me directly to the folder. And here I can see all of the artifacts that I'll want to use for my dartR analysis. Things like silico dart calls, snip calls. And this report here is in PDF form, is a publication-ready, human-readable report of similar to what Dart's applied to all of the customers for something like dartSec, and that will look something like this. Incidentally, all of the reports like this can be customized within our reporting templating here. So for an organization, you can actually put your own letterheads and things like that on here. At this point, I will pass back to Luis.
Thank you, Ruben. Very nice. Yeah, so the Reporter app is an awesome application that lets you customize in thousands of ways any report. That we also have access to this Reporter app into dartR that I'll show you very quickly. So I'm going to share my screen. And your screen, yes. Okay, so here we are again in the dartR implementation in the OneDART platform. And it's really it has been a lot of work for all the team. And now we have an interactive version of dartR, including all the 200 functions of dartR and counting. And hopefully with all the help of the users that are attending to this workshop today, and during this week we are going to increase many more times the number of functions. So we have here the dashboard. We start in the dashboard. Here is where we can customize all our plots. For example, here with this, we can choose the different color palettes for the populations of the color for our populations, and these same colors will be used in all the plots in dartR. And the same with the different plot theme, and also with this color palette of this type of data. Also, we can choose different formats for our plots, PNG, TIFF, VMP. We can also choose the different sizes for our plots. We have a small size, for example, if you have a very large heat map of thousands of individuals, you might use here maybe a grid of 50 by 50. We also have the features to customize the resolution of the plot. Then we have to the left, we have different types of modules in dartR. We have in orange, we have the general functions that, for example, we have data overview, functions, exploration, data filtering. And then in green, we have more specialized functions, for example, for natural selection, genetic diversity, genetic differentiation. At the top of each one of the modules, we have 3 main management panels. The one to the left is where we load our data. We can load the data from our computer or from the OneDART platform. And here we have just to drag and drop our files. And of course, dartR and OneDART does not just allows you to analyze DART data. You can use 14 different formats for genetic data. So we Plink, BCF, structure, and many other formats. So you have just to drag and drop your dataset here, and it will magically read it by the application. And for this presentation, I'm going just to use the Platypus dataset. That is a small dataset that will allow us to run very, very quickly the analysis. Also, we can load in the same way. We can load the metadata either from a CSV file or from the OneDART platform directly from EcoLog. Here in the middle, we have the current dataset panel, in which we have the updated information of our dataset. So every time we update or modify our GenLine object, this information will be updated. Then we have this bottom icon to check the individual metrics of our dataset. And here in this window, we can sort, we can delete rows, and we can modify and edit any of the fields and any of the cells, and we can search and we can download these individual metrics. And in the same way, we can use the same for the locus metrics. Here is all the information for each one of the locus, which and at this time also is searching across tens of thousands of loci. This search box makes everything easier. Then in this table, we have a table with the populations that we can also sort. And for example, if we want to remove Tenterfield, for example, we just select it and drop it. And in the right side, we have the dataset options panel, in which we can perform very actions that are we usually do often. For example, assigning population. If we want just to assign the sex as the population, and then our dataset the population will be, in this example, females and males. If we have information from our reference genome, we can assign the chromosome to each one of the loci, and we can also assign the SNP position. And this opens a wide windows of many more analyses that we can access. For example, if we are just starting to analyze our data, we can subset our data. So in this exploratory stage. And then we can hide this management panel to have more room for the analysis. And for example, we can go and get a sample locations. So in general, we have two parts in each one of the functions in dartR in this implementation. In the left, we have the description with the purpose, the methods, the guidelines, the output, and further reading for the function. We have an execution time approximation that changes depending on the analysis and the number of individuals, number of parameter values, and number of loci in the dataset. And then we run this analysis. We can send it to a PDF report. We can zoom in, and we can download it. Here we can generate a PDF report. And the most interesting thing is, or one of the important features of this implementation of dartR is that whatever we do in the application, everything is going to be translated into code. So we have just to copy this code and paste it in a RStudio session to reproduce everything we did in the platform. And yes, I think that's it for me for the moment. I hope you will enjoy it, and in the future we will add many more features, like together with our AI team. Yes. Okay. I guess we run out of time, and maybe I can go through some details of the last details that I want to share with you. For example, so I can show you how to find some things. I'm going to share my screen. Okay, you can see my screen now. I hope you can recognize this part. Here in the resources, we have shared tons of different resources that we have gathered during the workshop, including the presentations, the slide presentations of the presenters. And actually, Arthur is going to talk about this important document right here. So you will have access to these presentations and resources whenever you want. The ERMIT platform will be available for the next 3 months. And also, if you want to go and rewatch a session, you go to the mission log, either here or here, and then you go to a specific day. And we want, for example, we want to go to the very first session. We click here, and you will see watch replay. Okay, what other thing? Also, don't worry if all the slides of if not all the presentation, the slides of every presenter is not here in the resources. We are going to send all of them in probably next week. Okay, and also the POSIT Cloud will be available also for you to use for the next couple of weeks. And then also, if you look at...
Luis, can I just interrupt to explain the process? Because the POSIT Cloud is very simple. As long as you keep your account, you will have access to all the machines you copied. So make sure you copied the last one. The whatever 13 to 17 has all the code and everything, the latest one. And after a month or so, you will be defaulted to a normal account, which means you have only 25 hours compute time, but you can still log in, download everything, and even run it, but not more than 25 hours compute time. So there's no rush, but you can export. You will find it, there's an export button, and download the whole sessions in one go. And then you only need to have to install all the packages, which can be a pain, I admit that, but the data and the code you have will be there. Luis, you're muted. Sorry. You are still muted. Okay, I go on.
Sorry. Yeah. Here we are. So
dang, I thought I had my chance now. Huh?
You can see that during this workshop we used 15,000 hours of computation time between all the participants. So which is kind of nice. Okay, so then if you go here to the resources, you will find a very useful links, including the GitHub repository, where you can find all the code from this workshop. You can here's the link of the tutorial website, our YouTube channel, where we are going to upload all the speakers' recordings forever. And we have the YouTube channel of DART. We have our Google Groups forum link and our official website of dartR.
Luis, can I wrap for another 17 seconds?
Yes.
I have 2 polls because I like the polls, and because I'm a data-driven person, here is the first one. I just want to have a quick overview what people think about the air meet thingy thing, because we actually paid for that. You wouldn't believe it. Yeah, okay, we can. Cool. No, I think the numbers are kind of stabilizing. Yeah, maybe was okay. It's winning a little bit, gaining a little bit. Okay, so that's cool to see. I think, at least I take it as a positive. And I have one more, and then because that's what I want to know. I stop now. Ah, dang. Yeah. No, at the moment there's no terrible. That's because people are kind and wanna stop us. Okay. And I admit, never want to leave is a hard one. Okay, thanks a lot. That's actually, I take it as a compliment. Okay, thank you.
Okay, and one last thing from my part. Here we have, yes, so somehow this Hermit platform record every action we all did during the interaction we all did during the platform during the workshop, and there is actually a leaderboard. We can see that Samuel Ramellini from ANU was the most interactive person during the workshop. Then we have Josephine in second from Adelaide Senior Geneticist. And third one, we have Nalonda from UWA. And you can find your place in this leaderboard. And finally, we got 173 attendees in this workshop, which is great. And I guess...
Lewis, before we get too far from the DART side of things, did you want to check in with Andrzej?
Yeah. Yes, Andrzej is going to give the closing speech of the workshop.
Thank you. Sorry. Yeah.
Okay. I guess my last word is that I really enjoy the workshop, and I put a lot of effort, even if
It's
that
fine.
doesn't.
Okay.
If that doesn't was very important anyway. But thank you very much for all the speakers and the hard work of everyone. We are really, truly grateful for all your effort, time, and support. And now, Bernd, do you want to go?
I want to go. No. I want to I call all speakers, please, to the stage. Or is there anything I missed? I haven't told anyone. And please, switch on your screen. That would be nice. You know, my impression, because it's still recorded, we all wave and say goodbye, and then we close the final session. We don't have to sing anything, or do we? No, it's okay. And yeah, so are we all here? Is it all there? Hey, Florian, where are you? Where's your camera? Okay. Cool. I guess, yeah. Arthur wanted to say something, didn't he?
He's after you.
I'm after you, Bernd. and then Andrzej, right? So,
yes.
Yeah. So
Okay, I have nothing to say. I will thank everyone for being so kind and friendly and useful, and it was a pleasure to hold this stage, and would like to thank all the presenters, especially the one which is still with me here in the room. But yeah, thanks for everyone who is being involved, and I pass on to Arthur.
Okay, so basically, when I opened the workshop, I guess I raised everyone's expectations as to what the workshop would be like and how much value it would be. And I've got to say, from my perspective, it exceeded those expectations. It's been a great workshop, and I hope everyone agrees with me that it has exceeded expectations. There's been some wonderful talks and some excellent case studies. And we've moved from theory to practice and back to theory. We've moved from chalk and talk to hands-on to chat rooms. It's been a very interactive workshop, and all credit to Luis and Bernd for picking the platform in which to deliver this workshop. We've seen new tools available through dartRverse and also through OneDArT. And the OneDART's particularly exciting because it provides new coordinated opportunities for sharing data and for collaboration, which I think are going to be of real value and very exciting in the future. We heard about exciting new developments in AI, and I'd just like to draw your attention to the document I put up in the resources called AI on Speed. And I think if you read it, I reckon that your life will never be the same. So it's really worth having a look at. One prompt to an AI agent can generate a full analysis. Now, of course, you've got to validate that and be careful about your prompt, but man, is it an exciting tool. So now let's raise their emojis to thank Luis and Bernd for their Herculean efforts in making this workshop a success. I guess that's Bernd clapping himself there. And also to Ching Ching and Ethan for working tirelessly behind the scenes. I mean, they've been outstanding in their support. And thanks also to all the speakers for generously making their time available. And thanks to our sponsors, DART, CSIRO, and the Centre for Biodiversity Analysis. And I particularly like to identify Andrzej in DART, whose generosity and open collaboration has enabled dartRverse to flourish. And also, it's great to see it integrated into OneDART in the way it has been. So that's really good, Andrzej, and we're all very grateful to you. Now, Luis will be sending out a survey asking how the workshop went for you, and you'll have an opportunity to put suggestions for improvement in there. And we've got tough skins. So, you know, if you think it can be improved in some way, don't hold back. We're really interested in critique, and we'll take that on board to improve the next workshop. So finally, thanks again to all the participants and their engagement with us in this learning event. And I'm sure you all got as much out of this workshop as I did. And I'll pass over now to Andrzej to close the workshop.
Thank you. Can you hear me? Yes, okay, because on my screen it looks like my camera is off and I'm muted, but I'm glad that it's not the case. And I was a bit speechless for a moment when you, Arthur, showed that my small generosity is multiplied many times by other people. It's the lesson of my life. And it is always an emotional moment, and sometimes even sad when things come to close, like closing the workshop, but every ending is a new beginning. And in this case, I would hope that for you, it is an improved capacity to conduct more powerful research, contribute to protection of certain species, improve important ecosystems, or help humanity to eliminate hunger with the tools and knowledge that you gain through this workshop. We wish you great success in all your endeavors. My only remaining task is to say thank you to many people, and I'll be repeating after Arthur, great leadership, thank you, and you scooped a lot of things, but most importantly, I think to all participants who allocated their precious time to learn something new. And learning new things is the most precious things you can do. And I can assure you that as someone coming close to retirement, the older you get, the more important is it that you learn every day something new. And I trust that each of you has taken something useful and productive out of the workshop. Second, thank you very much to all the speakers. I'm sure that you all put a lot of effort preparing interesting content and delivering it in a professional and often exciting manner. That excludes my own, but well, I alluded to the aging process before. That definitely includes a DART team who not only just write a good demo for OneDART platform, but spend a lot of time preparing it, and many years in preparing OneDART platform. We hopefully will release it very soon to your benefit and beyond. Third, shout out to sponsors, CSIRO, Center for Biodiversity Analysis. I think Eureka Prize, I understand that they chipped in, not only recognizing the great job of dartR team, but also contributing to in some financial way to this workshop, and also our organization that put quite a bit of effort in it as well. So finally, let's acknowledge again, truly amazing, on maybe in the spirit of this workshop, transgalactic effort of people who spent months preparing and running this workshop. And Luis and Bernd and Ching Ching and Ethan have to be named in that context. So it is late afternoon, and I'm sure those in Kualoa are very eager to start some cold drinks, and those across Australia and across the world need to go back to their lives. Please stay in touch. We would love to support your efforts, either through provision of genomic service, help of data management or analysis, and whatever you do, enjoy it, do well, and take care. And see you next time. Thanks.
Great.